the grade 10 unit 6 exercise. This is the exercise from your textbook. The first question, as you can see, they have given the diagrams of two different cells. Name A, B, C, D, E structures and organelles in the above cells. So immediately when you look at the two cells, you should be able to differentiate and identify what type of cells they are. Here you can see it has a rectangular structure and also you can prominently see the cell wall. So you know this is a plant cell. And in this picture, you can see only one boundary or one layer around the cell. That is the cell membrane. So from that, you can easily identify this is an animal cell. Apart from that, you all know other differences that can be used to differentiate or identify a plant cell from an animal cell. Now let us identify all the organelles shown in these two diagrams. I am sure students, you have already identified the organelles and you have written down the answers. I will start off with the first structure, that is A. That you can see it's the outermost boundary in this cell and we already identified it as a plant cell. Therefore, A has to be the cell wall. So, A here is the cell wall. Then we have the second structure B. That is the next layer inside the cell wall. That is the second boundary that is present inside the cell wall. So that has to be the cell membrane or the plasma membrane. So here this will be cell or plasma membrane. Then we have structure C. That actually points to the space in between. You all know in a cell, all the organelles are present in the cytoplasm. So, C has to be the cytoplasm. Then we have D. You have identified the structure like a stack of saucers arranged one on top of the other, membrane-bound sacs arranged together with vesicles on the side. Here you can see the membrane-bound sacs and the vesicles there. So this has to be the Golgi complex. Golgi complex. Then we have structure E. Here you can see one structure here and another structure there. And they are large and prominent. So those are the vacuoles. Or we can say central vacuoles. Normally there is a very large central vacuole. Here there are two central vacuoles. Then we have structure F. Here you can see the structure F is part of the nuclear envelope or it actually extends from the nuclear envelope and also there are dots seen on the surface. You know what those dots are? Those are the ribosomes. So what is this folded membrane-like structure or the network of membrane? It is the rough endoplasmic reticulum. So here this is the rough endoplasmic reticulum. Then we have to identify G. Here you can see there are two structures which are similar, and in this one you can see this and that they have that folded membrane, and these two have like plate-like or lines in between them. So how do we differentiate? When there is a folded membrane, it has to be the mitochondria. And the other structure which has line-like structures inside will be the chloroplast. So here we can see G is mitochondria. So G, mitochondria. And H points to the dots in those cytoplasm. If there are very tiny dot-like structures, those are the ribosomes. Yes. And then we have I. I points to this organelle, and we have already identified it as the chloroplast. So I is chloroplast. And then we have to identify J and K. Here you can see this whole thing is the nucleus, and in that the structure inside is the nucleolus, and this outer layer is the nuclear envelope. Both of those together, in addition to that, there has to be chromatin body. All those together is the nucleus. So in this, J is the nucleolus and K is the nuclear envelope. So those are the structures and organelles shown in this diagram. I am sure you identified them correctly. Now let us move on to the next diagram. So as we identified it before, this is an animal cell. No cell wall present. The outermost boundary is the cell membrane or the plasma membrane. So here you can see structure A is the cell membrane or plasma membrane. Then we have B. Again here you can see this is B. It points to the space there. So the space, that is the gelatinous structure that contains all the organelles. What is that? It is the cytoplasm. So B is the cytoplasm. Then we have C. Again similar to the previous diagram, membrane-bound sacs arranged one on top of the other with vesicles around it is the Golgi complex. So C is Golgi complex. Golgi complex. And then we have D. Here you can see D points to those small dots. Those are the membraneless structures, organelles that do not have a membrane, and they are the ribosomes. Ribosomes. Structure E. Here you can see structure E. And structure E is a membranous network. And in that, around the membrane, you can see the dots. These dots are the ribosomes. So, there are two types of membranous network. The rough endoplasmic reticulum that has the ribosome and the smooth endoplasmic reticulum that doesn't have the ribosome. What is E? It is the rough endoplasmic reticulum. So, here it is the rough endoplasmic reticulum. Then we need to identify F. So, as you can see in the diagram, F and G are parts of the nucleus. What is F? It is the nuclear envelope, the outer membrane of the nucleus, and inside we have the nucleolus. So, F is the nuclear envelope and G is the nucleolus. Then we have structure H. What is structure H? It is a small vesicle-like structure present throughout the cytoplasm, and that is the centriole. So, here we have centriole. And the last structure, that is I. This is an organelle that has two membranes around it and is known as the powerhouse of the cell. What is it? It's the mitochondria. So, I is mitochondria. So, I am sure students, you identified all these structures and the organelles correctly. Let us now move on to the next question. Differentiate between a plant cell and an animal cell. This is something you know very well. So, let me do it in the form of a table. I will put the two headings, plant cell, and on this side we can have the animal cell. Then we will start off with the outermost layer. What is it? The cell wall. It is present in the plant cell. So, cell wall is present. Here it is absent. What is the second difference? There is a large central vacuole in the plant cell and there is no large central vacuole in the animal cell, but there can be small vacuoles present in an animal cell. So, the second difference, presence of large central vacuole. Here absent or small vacuoles are present. So, because of this large central vacuole, what happens in the plant cell? Cytoplasm is towards the periphery and also nucleus is pushed towards the periphery. So, the next one, cytoplasm is pushed to the periphery. Here it is cytoplasm is spread throughout the cell. The next one, nucleus is in the periphery. Nucleus is in the periphery. Or I can say nucleus is pushed to the periphery. And in an animal cell, nucleus is centralized. Nucleus is centralized. And what is the last difference? In a plant cell, there is chloroplast. But in the animal cell, it is absent. Yes. So, chloroplast is present. In an animal cell, it is absent. So, students, now these are the basic differences between a plant cell and an animal cell. So, if they ask you to differentiate between the plant and animal cell, you have to write everything. Otherwise, when they give you the diagram and ask you to identify the cells, and if they ask you what character of property that you use to identify the different cells, then you will have to write the property that you used. Maybe from the presence and absence of a cell wall, you differentiated the two cells. Then you can write that. So, depending on the question, you have to write the answer. And if they ask for two differences, then you can write any two differences. With that, we will move on to the next question. Mention the functions of the following organelles. Under that, the first one is mitochondria. You know the special name of mitochondria? It's the powerhouse. Why? Because the aerobic respiration takes place in the mitochondria. So, the function is aerobic respiration. Respiration produces energy. Then we have Golgi Complex. What is the function of Golgi Complex? It produces secretory substances, packaging of secretory substances, and secreting the substances. So, here production of secretory packing and secretion. Then we have rough endoplasmic reticulum. Why do we call it rough endoplasmic reticulum? Because there are ribosomes on the membrane of the endoplasmic reticulum. What does the ribosome do? They produce protein, or they do protein synthesis. So, what does the rough endoplasmic reticulum do? it transports the protein. So, rough endoplasmic reticulum, transport of protein. Then we have vacuole. The large vacuole, what does it do? It stores or contains water, ions and pigments. Therefore, it gives a shape to the cell or helps to maintain the shape of the cell. It helps in water balance of the cell and also it gives a color to the cell. So, here they have us mentioned the functions of the following organelles. So, I will write down all the functions there. So, here first one is contains water, iron and pigments. Therefore, it helps in water balance. Water balance. Also, it helps to maintain the shape of the cell. And finally, it gives color to the cell. Provides or provision of color. So, those are the functions of the vacuole. We will move on to the next slide. Explain the importance of meiosis. We have discussed both the types of cell division, and in that meiosis is where the number of chromosomes become half. The mother cell is diploid and the daughter cells are haploid, and this helps to maintain the number of chromosomes from generation to generation in a species, or to maintain the number of chromosomes of a species. So, here one importance is helps to maintain the number of chromosomes specific to a species from generation to generation. What is the next significance? There are changes occurring to the chromosomes. That is variation. Therefore, new characteristics are formed, and that is important for evolution. So, the second one is variations take place during meiosis. This is important in evolution. So, with this, students, we have finished answering all the questions in your textbook. Now I have added some extra questions. What you can do is, when I read the question, you can pause the video, you can answer the questions first and then listen to my discussion to confirm that you have written the correct answer. So let us move on to those questions. Extra question 1. All living organisms are made up of one or many cells. Diagrams A and B given below are two types of cells taken from living organisms. So, as you look at the diagram, first you should identify whether it's observed or seen through a light microscope or whether it's an electron microscope image, which gives all the organelles and structures present. Here you can see it's just the simple cell diagram. So, these are cells observed through the light microscope. And here you can see there is only one boundary around the cell and there are nucleus almost towards the center of the cell. Then you know these are animal cells. If you look at cell that is present as B, there are two layers that are there as the boundary. So that means that is the cell wall and you can see the nucleus towards the periphery of the cell. So you can confirm that they are plant cells. So after identifying that, if you read the rest of the questions, identify the animal cell from above. What sample can you use to observe cell A? Identify and name the organelles given below. Here the diagrams are given. I am very sure that you can identify the organelles easily. Write the functions of organelles X and Y. What is the main component that make up the cell wall? Write two features present in a plant cell but are not present in an animal cell. Write two contents of cell theory. So once you finish answering the question, you can listen to the discussion. I will move on to the answers. All living organisms are made up of one or many cells. Diagrams A and B given below are two types of cells taken from living organisms. We identified the two cells, A as an animal cell, Y because it has only one boundary, that is the plasma membrane, and a centralized nucleus. And B as plant cells because they have a cell wall and the nucleus is pushed towards the periphery of the cell. Identify the animal cell from above. So that is cell A. What sample can you use to observe cell A? You can remember the activity, students. What do we use? The cheek cells. So here you can say a sample of cheek cells. I will move on to the next question. Identify and name the organelles given below. So we have three organelles. What is the first one? That is X. How do you identify this organelle? It's a rod-shaped structure and you can see that there is a folded membrane inside. So that means it is the mitochondrion. Then we have organelle Y. You can see it has a large body and a small body. So that means it is a ribosome. And we finally have organelle Z. Very easy to identify. It has a membrane around it, a small circular structure inside. The membrane is the nuclear envelope. The circle inside is the nucleolus, and the thread-like structure inside is the chromatin body. So, Z is the nucleus. Okay, now the next question. Write the functions of organelles X and Y. We identified X as the mitochondrion. So here, if we write the function of mitochondrion, you know that very well. Aerobic respiration takes place in the mitochondrion and energy is released. So here we can say energy is released by aerobic respiration. That is the function of X. Then we have organelle Y. Just to show you all what organelles X and Y are, I come back to the slide. Here you can say X is mitochondrion, we identified that, and Y is ribosome. So if I go back to the slide, the function of organelle Y. You know the function of ribosome? Protein synthesis. Okay. Next question. What is the main component that make up the cell wall? I'm sure you remember your Unit 1 lesson, biomolecules, and also we discussed that component here. Cell wall mainly contains cellulose, which is a polysaccharide, a type of carbohydrate. So, what is the main component that make up the cell wall? You have to say cellulose. We will move on to the next question. Write two features present in a plant cell but are not present in an animal cell. So, there are many structures, or at least a few structures, that are absent in an animal cell and are present in a plant cell. What are they? The cell wall, large central vacuole, and the chloroplast. So, you can write any two features. I will write the cell wall and chloroplast. You can write any two structures. You can also write the large central vacuole there. Write two contents of cell theory. I'm sure you remember the cell theory, students. It was proposed by Schleiden, Schwann, and Rudolf Virchow. And there are three contents. Cell is the structural and functional unit of life. All organisms are made up of one or more cells, and new cells are formed from pre-existing cells. You can write any two contents. So, I will write, so cell is the structural and functional unit of life. That is the first content. Then I will say new cells are formed from pre-existing cells. So those are the two contents I have written. You can also include that all organisms are made up of one or more cells. We will move on to the next question. Extra question 2. Given below is part of a diagram of a typical cell. I am sure you all know what a typical cell is. A model of a cell made with all the organelles that can be present. The rest of the diagram had been torn off. So here it's only a part of the diagram. The rest of the diagram is missing. What type of cell can you see in the above diagram? Write the feature that helped you to identify the cell given above. Write an important feature of the cell that is not seen in the diagram above, but can be seen in the torn part of the diagram. Write a function of the organelle you mentioned in part 3 above. Now, you all answer the questions and then we will discuss it. Given below is part of a diagram of a typical cell. The rest of the diagram had been torn off. And the first question is, what type of cell can you see in the above diagram? So here students, you can see there is only one line as the outer boundary. That is a feature of an animal cell because there is no cell wall. So that is one feature that you can use to identify the cell. Then here you can see there are mitochondria present. And what is this structure? It is a centralized nucleus. So you can see only a part of the nucleus. But you cannot confirm that there is no large central vacuole because that can be present in the other part of the diagram. So here we use the presence of only one border, that is the plasma membrane or cell membrane, and the presence of a large nucleus to identify the type of cell in the diagram. So what type of cell can you see in the above diagram? It is an animal cell. Animal cell. Write the feature that helped you to identify the cell given above. Presence of only the cell membrane. So here you can say presence of cell membrane only. No cell wall. Both help you to identify that this is an animal cell. The next question, write an important feature of the cell that is not seen in the diagram above but can be seen in the torn part of the diagram. So here a part of the cell diagram is given to you. Nucleus is there. Mitochondria is there, and endoplasmic reticulum is also there. What is the organelle that is missing? Yes, it is the Golgi complex. So you cannot see the Golgi complex here. Write a function of the organelle you mentioned in part 3 above. What is the function of a Golgi complex? Production of secretory substance, packaging, and secretion. So here you have to write production of secretory substance, then packing and secretion. So those are the 3 functions of Golgi complex. Now we will move on to the next question. Extra question 3. Figure 1 and 2 given below are diagrams of a plant and an animal cell drawn based on information obtained using the electron microscope. Here you can see figure 1 and figure 2. Identify the diagrams and write in the lines provided below. So you have to identify the 2 types of cells, figure 1 and figure 2. As shown in the diagram above, write 2 features seen in the plant cell but not in the animal cell. Identify the structures labeled as X and Y. Write 1 function of each structure. Write the name of the organelle known as powerhouse of the cell. Briefly explain the reason for this name. Write 2 structures that can be observed in a plant cell through the light microscope. Write 3 steps involved in the preparation of a temporary slide of an onion peel to be observed under the light microscope. I am sure you can answer all the questions by yourselves first. So do that and then listen to my discussion. Figure 1 and 2 given below are diagrams of a plant and animal cell drawn based on information obtained using the electron microscope. I am sure you know, students, when we observe the cells under the electron microscope, we can see and get information related to the organelles present. So this structure shows us most of the organelles present in a plant cell and an animal cell. So what do we have to do first? We need to identify the 2 cells. If you look at figure 1, first start off with the outermost boundary. Here you can see there are 2 lines, and here you can see there is only 1 line, only 1 layer. That means figure 1 is a plant cell and figure 2 is an animal cell. We can further confirm that by looking at this structure, the central vacuole and the nucleus being at the periphery, whereas in figure 2, there is no large central vacuole and there is a centralized nucleus. So we can confirm and say that figure 1 is a plant cell and figure 2 is an animal cell. Identify the diagrams and write in the lines provided below. So you have to write that here again. Figure 1 is plant cell, and figure 2 animal cell. The next question. As shown in the diagram above, write 2 features seen in the plant cell but not in the animal cell. So, we can write down that there is a cell wall and large central vacuole, and also here you can see structure X that is specific only to plant cells. What is that? Chloroplast. So we have seen 3 structures. You can write any 2 structures that are present in plant cell but absent in the animal cell. So I will say large central vacuole and chloroplast. You can also mention the cell wall. Next one. Identify the structures labeled as X and Y. Write one function of each structure. We have already identified X. What is it? It is the chloroplast. So here I will say X, chloroplast. And what is the function of a chloroplast? Photosynthesis. So here I can say photosynthesis. Next we have structure Y that is shown in figure 2. Can you identify this structure, students? How does it look like? Membrane sacs arranged one on top of each other. So these are membrane bound sacs arranged together, and also here you can see few vesicles around it. So what is that structure? It is a Golgi complex. So here I will write structure Y as Golgi complex. And what is the function of Golgi complex? Production of secretory substances, packing and secretion. So here I can write production of secretory substances, packing and secretion. So those are the functions of Golgi complex. We will move on to the next part of the question. Write the name of the organelle known as powerhouse of the cell. Briefly explain the reason for this name. What is known as the powerhouse? The mitochondria. So here we will say mitochondria. And why do we call it the powerhouse? Because energy is produced during aerobic respiration that takes place in mitochondria. So here I can say energy is produced by the aerobic respiration that takes place in mitochondria. And this is the energy that is given for all the cellular processes. That is why we call it the powerhouse of the cell. I'm sure you got that right. Then the next question, write two structures that can be observed in a plant cell through the light microscope. What are the structures you can see? You can see the cell wall and you can see the nucleus because it is through the light microscope. But you know, students, usually the plant cells are green in color when you look through the microscope. Why? Because there is chloroplast or chlorophyll present. But we can't really see the chloroplast clearly. For that, we need to use the electron microscope. So here I can say we can see the cell wall and the nucleus. We will move on to the next question. Write 3 steps involved in the preparation of a temporary slide of an onion peel to be observed under the light microscope. We have done this activity before. You know the steps involved. What do we do first? We peel the onion, remove the skin and take out a fleshy part of the onion peel. From that, we cut very tiny specimens or peels from the flesh and put that into either a watch glass or a petri dish containing water. Then we take a clean glass slide, put a drop of water on the glass slide using a paintbrush and transfer a very thin peel onto the glass slide. After that, what do? We cover it with a cover slip without trapping any air bubbles. Then the slide will be ready for observation. And what is the name given to that slide? We call it the temporary slide. So we need to write down the preparation of a temporary slide in 3 steps. So the first step, I will say, peel the skin of the onion and remove a fleshy part. Then we can say the second step, cut very thin peels from the fleshy part and put into water. What is the third step? Put a drop of water on a clean glass slide. Place a thin peel using a paint brush and cover with the cover slip. So here we have given the method of preparing temporary slides in 3 steps. I'm sure you got this answer also correct. So let us move on to the next question. Extra question 4. The cell theory proposed based on the facts revealed by observing different live tissues through the microscope is very important in biology. That you already know. Name the scientist who proposed the cell theory. Who are the scientists? I'm sure you know the answer for this. Schleiden, Schwann and Rudolf Virchow. So I'll write their names. Schleiden, Schwann and Rudolf Virchow. So these are the 3 scientists who proposed the cell theory. Write 2 contents of cell theory. You know that there are 3 contents. Cell is the basic structural and functional unit of life. All organisms are made up of 1 or more cells, and new cells are formed from pre-existing cells. We can write any 2 contents. I will say cell is the structural and functional unit of life. And the second one, I will say all organisms are made of 1 or more cells. You can also write the third content. New cells are formed from pre-existing cells. Then the next question. Specify a sample that could be used to observe the animal cell in the classroom. We have done this activity before. You know the sample that we used. It is the cheek cells. So here you can say a sample of cheek cells. So I'm sure you were able to answer all these questions. Now I will move on to the next question. Extra question 5. Diagram of an animal cell observed through an electron microscope is given below. You can see the diagram there. Write the functions of the structures A, B and C. They are shown in the diagram. Write 2 structures that are not present in this cell but are present in a plant cell. New cells are formed from the division of the above cell. Name the 2 methods by which nucleus divides. In one of the methods you mentioned in part 3 above, only half the number of chromosomes from the mother cell are given to the daughter cell. Name that cell division. What is the cell division that takes place when a wound is healing? I am sure you can answer these questions by yourself first. Now, let me move on to the discussion. Diagram of an animal cell observed through an electron microscope is given below. So, here you can see it's an animal cell, and also because it's the electron microscope image, you can see most of the organelles. Write the functions of the structures A, B and C. So, in this diagram, you can see what A is, this structure. You are very familiar with that structure. What is it? It is a Golgi complex. And how did you identify it? From the presence of the membrane-bound sacs arranged one on top of each other and the vesicles present around it. So, A is Golgi complex. So, I will write A here, but here they have only asked for the functions of structures A, B and C. So, you don't have to write the name Golgi complex, but you have to say the function of a Golgi complex. There are 3 functions. I am sure you know them very well. Production of secretory substances, packing and secretion. So, that is what we have to write here. Production of secretory substances, packing and secretion. Then we have structure B. What is structure B? You can easily identify it as the nucleus. And what is the function of a nucleus? It controls the activities of the cell. So, here we can say controls the activities of the cell. Then we have structure C. Can you see the structure C here? Yes, it is a rod-shaped structure with the folded membrane inside. So, what is it? It's the mitochondria. And you know the function of the mitochondria. Aerobic respiration. So, C, production of energy by aerobic respiration. So, we have written down the functions of structures A, B and C. Let's move on to the next question. Write 2 structures that are not present in this cell but are present in a plant cell. So, here the 2 structures have to be absent but should be present in a plant cell. There are 3 structures. One is the cell wall, the other one is the chloroplast, and the third one is the large central vacuole. You can write any 2 structures. So, here I will write the structures as cell wall and large central vacuole. New cells are formed from the division of the above cell. Name the two methods by which the nucleus divides. You know the answer very well. The two methods, mitosis and meiosis. So here we can write mitosis and meiosis. Next question. In one of the methods you mentioned in part 3 above, only half the number of chromosomes from the mother cell are given to the daughter cell. Name that cell division. In what cell division is the number of chromosomes halved? The meiosis. So the answer will be meiosis. What is the cell division that takes place when a wound is healing? When a wound is healing, identical cells have to be produced from the existing cells. Then the cell division has to be mitosis. So here this has to be mitosis. I am sure you knew all the answers to the questions. Now let us move on to the next question. Extra question 6. Diagram of a plant cell observed through an electron microscope is given below. So here you can see, students, the structure of the plant cell with the different organelles marked with English alphabets. First question, write the letters A, B, C, D and E that denote the structures given below. Nucleus, chloroplast, Golgi complex and mitochondria. Write the letters relevant to the organelles that carry out the production of secretory substances and photosynthesis in order. Write the name of the main structural component present in F. Compare the permeability of structures F and G. I am sure you will be able to answer all these questions. Once you are done, we will discuss the answers. Diagram of a plant cell observed through an electron microscope is given below. Write the letters A, B, C, D and E that denote the structures given below. So the first structure is nucleus. Out of the letters given here, observe the diagram. You can easily identify the nucleus. Here you can see the nuclear envelope, chromatin body and the nucleolus. So structure D is nucleus. So here you just need to write down the letter. Then the next one is chloroplast. How do we identify the chloroplast? There are two structures which can confuse you, C and B. They both look like rod-shaped structures. But in B, you can see the folded membrane inside. So that is a mitochondria. Whereas C has these line-like structures or plate-like structures in between. So that is chloroplast. So structure C is the chloroplast. Then we have the third one, Golgi complex. You are very familiar with this structure. These are membrane-bound sacs arranged one on top of each other. Is that all? No. There are many vesicles around the membrane-bound structure. So here try to find a structure like that. You can see structure A, the membrane-bound sacs and the vesicles around it. So it will be structure A. And finally, mitochondria. You know mitochondrion is 1 Mitochondria, many. So there are more than one mitochondrion shown here, and that is denoted by letter B. So we have identified that already. B is mitochondria. Let us move on to the next question. Write the letters relevant to the organelles that carry out the production of secretory substances and photosynthesis in order. What is the organelle that produces secretory substances? Golgi complex. And in which organelle does photosynthesis take place? Chloroplast. So we have to identify Golgi complex, which we have already done, A, and chloroplast is C. So we have to write the letters in order. You have to write it as A, C. Write the name of the main structural component present in F. If you look at F, that is the outermost boundary, and it was told to us that this is the plant cell, so that is the cell wall. And in the cell wall, what is the main component? You all know it is a carbohydrate and it's a polysaccharide. The component is cellulose. So here we write the component as cellulose. Next question. Compare the permeability of structures F and G. So here from the diagram we have already identified F. It is the cell wall. And what is G? It is the second layer inside the cell wall. Second boundary that is present for the cell. And that is the cell membrane or the plasma membrane. What is the main difference in permeability? The cell wall allows all the substances to enter into the cell. So that means it's completely permeable. Whereas the cell membrane, that is G, that does not allow all the substances to come in or go out of the cell. It selectively allows certain substances. For example, water, some ions, and a few other compounds are allowed to come into the cell or go out of the cell through the cell membrane. So therefore, that is selectively permeable. So here we are supposed to compare the permeability of structures F and G. So here I can say F, cell wall, is completely permeable. And G is the cell membrane is selectively permeable. So that is enough to compare the permeability of cell wall and cell membrane, that is structures F and G. Now let us move on to the next question. Extra question 7. Underline the correct answer. So you know when we say underline the correct answer, there are 4 answers. Multiple choice type questions, MCQ questions. The first one, A, B and C given in the diagram below are. So they have given you diagrams. You have to identify the structures. A, you know this very well. A rod-shaped structure and you can see the folded membrane inside. So it is a mitochondrion. Look at structure B. What is that? Membrane-bound sacs stacked one on top of the other with the vesicles around it. So that is the Golgi body or the Golgi complex. And C, can you identify C? It has two bodies, a large subunit and a small subunit. So what is that? That is a ribosome. So we have to select the answer that contains the correct order of organelles. Ribosome, Golgi complex, mitochondrion. No. Mitochondrion, ribosome, Golgi complex. No. Golgi complex, ribosome, mitochondrion. All three have wrong orders. Mitochondrion, Golgi complex and ribosome. The fourth combination is the correct order. So this is the correct answer. So I will move on to the next question. What is the semi-permeable membrane of the cell? You all know in the animal cell there is only one cell membrane. Plant cell has the cell wall and the cell membrane or the plasma membrane. And out of those two, the cell wall is completely permeable, but the cell membrane or the plasma membrane is semi-permeable. Same in the animal cell, the cell membrane or the plasma membrane is semi-permeable. Let us look at the answers. Cytoplasm, not a membrane. Plasma membrane, yes, that's the correct answer. Cell wall is completely permeable. Nucleus, it is not a membrane. Third one. Pictures given below are two organelles present in a cell. Names of A and B respectively are. First one, you can see these membrane-bound sacs and the vesicles. So immediately you can identify it as the Golgi complex. And here you can see a network of membrane. Then you know it is the endoplasmic reticulum. But you have to look at the surface. Does it have ribosomes? Yes, that means it is rough endoplasmic reticulum. If there are no ribosomes, it would be smooth endoplasmic reticulum. Let us look at the answers. Mitochondria, Golgi complex. No. Golgi complex, rough endoplasmic reticulum. That's the answer. Just look at the other two answers. Nucleus, mitochondria, no. Endoplasmic reticulum, Golgi complex, no. So we'll move on to the next question. What is known as tonoplast? I'm sure you remember these, students. In the cell, there are different types of membranes. The cell membrane or the plasma membrane that surrounds the cell. Then you have different organelles having membranes. The mitochondrion, the chloroplast, all these are membrane-bound structures. Then the central vacuole. That has a membrane, and that membrane is known as the tonoplast. Now if we look at the answers, membrane surrounding the vacuole, that is the correct answer. Cell wall, no. Plasma membrane, nuclear envelope. Those are also membranes, but they are not called tonoplast. Fifth question. Organelles present in both plant and animal cells are plasma membrane, yes, and ribosome, yes. So that is a correct answer. Cell wall, not present in animal cells. Nucleus, yes. Chloroplast, only in plant cells and mitochondria in both cells. Vacuole and chloroplast both are present mainly in plant cells. Large vacuole in plant cell and chloroplast only in plant cells. So these three are not correct. The next question, irreversible increase of size or dry mass is known as, this is the definition for cell growth. Cell division, cell differentiation, cell growth, cell specialization. The answer is cell growth. You know what cell division is. Increasing the number of cells by the division of the nucleus and then the cytoplasm. Cell differentiation. All the cells are produced from one basic cell. That is the zygote that is produced by the fusion of egg and sperm. Those are the gamete cells. But from that, if we take the structure of our body, we have so many different types of cells. Cells of the skin, cells in the hair, the teeth, the different systems. So in all these, the cells become different because of their different functions. And that process is known as cell differentiation. And cell specialization, that is also the process of the cell becoming different and performing specific functions. So those two don't have this particular definition. Irreversible increase of size or dry mass is known as cell growth. Next one. What is a typical cell? Structural and functional unit of living organisms. All cells are that. Cell that produces another cell, we normally call it the mother cell. Cell that can undergo differentiation, usually that is the mature cell. Model of a cell that contains all the organelles. That is the meaning of a typical cell. So that is the answer. The next question. Which one of the organelles given below releases energy during aerobic respiration? You know the answer to this question. And that is called the powerhouse of the cell. What is it? It's the mitochondria. Nucleus, second one mitochondrion, the answer. Then ribosome, Golgi complex. Ninth one. Given below is an illustration of the formation of offspring from parents. You are familiar with the term offspring. Cell division that take place in cells A and B respectively are. Let us look at the diagram. The mother and father both have the cells. From them, the egg and sperm are produced. So then, what is the cell division? It has to be meiosis. Then only the number of chromosomes will become half. So here A will be meiosis. Then after egg and sperm combines, it's the zygote. From the zygote, the child will grow. The fetus will develop and the child will be born, and then thereafter the child will grow. So their zygote becomes the fetus due to mitosis, cell division. So A has to be meiosis, B has to be mitosis. So the correct combination here, first is mitosis and mitosis, no. Second is meiosis and meiosis, no. Third, mitosis and meiosis, no. And the fourth answer, meiosis and mitosis. That's the correct combination. We'll move on to the next one. Select the incorrect combination. the organelle and the function. Mitochondrial cellular respiration. That's correct. Chloroplast, synthesis of food. That is also correct. Why? Photosynthesis takes place in chloroplast and that leads to the production of food. Ribosome, production of secretory substances. That is not correct because in ribosomes, protein synthesis takes place. Rough endoplasmic reticulum, transportation of protein. That is also correct. So, the answer will be ribosome, production of secretory substances. Question 11. In what cell division can variations take place? This is something that you know, students. Only in meiosis variation will take place. Let us look at the answer. In mitosis only? No. In meiosis only? Correct. In both mitosis and meiosis? In neither mitosis nor meiosis. Both those answers are not correct. We will move on to the next one. The scientist who identified the cell is. You know the name of the scientist? There are 4 scientists involved with the cell and the structure or the cell theory. One person who identified it, he is Robert Hooke. And the 3 scientists who proposed the cell theory, Schleiden, Schwann and Rudolf Virchow. So, who is the scientist who identified the cell? That is Robert Hooke. And the others, Schleiden, Schwann and Virchow, they proposed the cell theory. So, students, with this question, I have come to the end of the discussion of Unit 6 of your Grade 10 Science textbook. I am sure you understood the structure of cells, the organelles present in both plant and animal cells, their functions, the cell division, cell growth, and why those are significant to us. As I always say, this is not enough. You have to watch the video again, get a better understanding, and also read your textbook so that you will become competent enough to answer any type of question that will be given to you.